Hello, everyone. Today we are going to talk about biomarkers from discovery to implementation. So, one of the very first questions that I want to ask you, Garima, is that mechanistic markers and on clinical biomarkers. Can you talk more on them and enlighten our audience?
So basically, you want to know about the difference between a mechanistic marker and clinical biomarker, right?
Exactly. Let's talk about that.
One distinction that really changed how I think about research is the difference between a mechanistic marker and a clinical actionable biomarker. See, at first they sound pretty similar. After all, both involve measuring something about the brain or behavior. But the more you sit with it, the clearer the difference becomes. So, a mechanistic marker, in simple terms, helps us understand how something works. So, for example, a behavioral task that captures fear learning can tell us a lot about threat processing. Similarly, a brain signal linked to an emotional regulation might help explain vulnerability to anxiety or PTSD. And don't get me wrong, that kind of work is essential, especially in psychiatry, where mechanisms are still very much an open question. But here's where things start to shift. A clinical biomarker isn't just about understanding, it's about decision making. It has to help answer questions such as who might be at risk, who is likely to respond to treatment, and is someone actually improving or not. So, while a mechanistic marker might work really well in a controlled lab setting, a clinical biomarker has to survive, real life, different people, different context, and a lot of noise. And that's often where the gap appears. Not because the science is weak, but because the clinical usefulness demands a very different level of reliability. So I think that is the main difference between a mechanistic marker and a clinical biomarker.
Right. Let's move into the direction of psychiatry and let's discuss about why psychiatry pushes us towards composite biomarkers, do you think?
Okay. So this gap becomes even more obvious when we look at psychiatric disorder. See, unlike some neurological conditions like Alzheimer's or Parkinson's, depression or PTSD don't have single clear biological signature. There isn't one brain reason, one gene, or one molecule that explains everything, which is the case of Alzheimer's or Parkinson's or any other neurological disorder. So naturally, researchers have started moving towards composite biomarkers. Instead of asking, where is the biomarker? The question becomes, what patterns of signal keeps showing up? Like what consistently keeps showing up? We are talking about consistency. For example, behavioral measures might capture avoidance or reward sensitivity. Brain data might reflect circuit level changes, and a physiological signal could tell us something about stress regulation. And genetic factors might indicate vulnerability rather than certainty. Each of these measures, that is behavioral, brain data, physiological, and genetic factors, on its own is pretty noisy. But when you put them together collectively, sometimes a more stable picture starts to emerge. What I find interesting is that behavior often ends up playing a central role. Behavior gives context to brain activity. It helps translate biological signals into something that actually reflects lived experience. And in a way, that's why behavioral neuroscience feels so important for psychiatry. It connects mechanism to meaning.
So, my follow up question would be if naturally we are moving towards a direction, let's suppose a person wants to pursue a project or a PhD in biomarker focused, style of that project or that angle of that project, then what should these the applicants keep in mind during these applications?
Okay. So now, if you're someone thinking about a PhD, which is where I am at, this naturally raises another question. Like, what actually makes a biomarker focused project strong? Early on, it's very tempting to think that strength comes from ambition, from saying you'll discover a new biomarker or fundamentally change diagnosis or treatment. And I completely understand that instinct. But the more time you spend with the literature, especially in psychiatric neuroscience, the more you realize that the strongest projects tend to be much more grounded. For instance, replication matters a lot. If an effect only appear once and it's interesting, so you can say that uh, just the effect appearing once and being interesting is not enough. It should be replicable further. Only then it makes sense. Similarly, effect size matters just as much as statistical significance. A result can be significant and still not be very useful. There are numbers of paper, which have proven to show significant results, but they are not applied or they are not very useful in real life. And last one I can say is there's robustness. Like it answers questions such as does the marker hold across different samples or tasks, or does it appear as soon as conditions change? In psychiatric research, variability isn't just noise, it's part of the story. So, thoughtful projects often try to understand when and why a marker works rather than assuming it always will. That approach to me feels much more closer to real translational science.
Right. So, I think there's this bottleneck issue where we always see there are so many biomarkers that we know in theory, but they never ever reach the clinic. Why do you think so that happens?
Okay, so before answering your question, I would like to share my own experience. When I started reading about biomarkers in neuroscience, I'll be honest, it felt a bit confusing. I mean, on one hand, everything sounded really exciting, but at the same time, everything also seemed to be called a biomarker. Brain scans were biomarker, behavioral tasks were biomarker, even stress hormones and genes were biomarkers. So, at some point, I had to stop and ask, if so many things count as biomarkers, why do so few of them actually make it into clinical use? And eventually, I realized even when a biomarker looks solid on paper, there's still another hurdle, that is the healthcare system. Yeah, we all know that clinicians are under time pressures and resources are limited as well. And any tool they use has to be relatively easy to explain to patients. So, a biomarker that requires complex analysis or expensive equipment, while scientifically impressive, may not be very practical. It seems very theoretical to have such ideal conditions, right? And on the other hand, simpler behavioral or physiological measures, especially when combined thoughtfully, often have a clearer path towards real world use. So translation, isn't just about better experiments. It's all about designing tools that fits into existing system. And that is what is missing, and hence so many biomarkers we know about them, but they don't reach clinics.
Wow, that was indeed a very well rounded explanation. So, we are coming towards the closing reflection. So, if you have any, please go ahead and share it with us.
Okay, so I would like to conclude by saying that when I think about why biomarker research matters to me personally, it really comes down to balance. On one side, there's the excitement of understanding how the brain and behavior work. On the other side, there's the responsibility to make that knowledge useful. Biomarker research sits right at that boundary. And as someone drawn to behavioral and psychiatric neuroscience, I'm less focused on finding a perfect signal and more interested in learning how to ask questions that actually translate. Because in the end, that's what decides whether research stays in general or whether it genuinely helps people. So, thank you for listening to me patiently.
Thank you, Garima, for all these informational insights. It's always been a treat to talk to you. Thank you. Thank you to the audience for listening to us.